Hello students, I welcome you all to my video lecture on node gene regulation and nodule formation. So in my previous class, we started with the study of symbiotic nitrogen fixation, taking an example of rhizobium species. Now, let us study in detail about the symbiotic association of rhizobium with that of leguminous plants and the genes which are involved in the formation of the nodules. So the first step involved in the process of nodule formation, as I was mentioning before also, is recognition. As we all also know that the host as well as rhizobium, they are species or strain or variety specific. They are specific to that of a particular host. So the rhizobium has to recognize the appropriate host. So this is done by the chemical signaling or the signaling molecules produced by that of a host plant as well as the rhizobium. So that process is called as recognition. Let us study those recognition, how actually the signaling transduction happen and all in my future slides. So the first step is recognition. Once the rhizobial cell recognizes the proper host, the next step is infection. The process of infection takes place by the particular substance which are produced by that of a rhizobium called as nod factors. This nod factors triggers the gene expression of that of a host, so that the infection or the entry of the rhizobium inside the host cell become easier. So the next step of the nodule formation is the penetration or entry of the rhizobium inside the host by the formation of infection thread. Once the rhizobium come in contact, once it recognizes the host, once it come in contact with that of a root hair of the host, they attach onto the surface of the cell of the root hair. There are certain substances or the recognition sites which are present on the host as well as on the rhizobium. Rhizobium produces a particular substance called as Rick adenosine. Through this, with the help of this Rick adenosine, it's a calcium binding protein where this attaches, rhizobium attaches onto the surface of the host cell on the surface of the root hair with the presence of Rick adenosine on its surface. And also the root hair also has got a particular recognition sites which are called as lectins. So the Rick adenosine as well as the lectin, they help in the process of attachment of this rhizobium onto the root hair. Once it attaches, at the same time the cellulosic thread or the infection thread or the cellulosic tube is being produced by the host. Through this cellulosic tube, the rhizobium proliferates and they cause infection and they enter into the host tissue through this cellulosic or infection thread. So the third step is infection. The first step was recognition, the second step was attachment, the third step of the formation of the root nodule is infection thread formation and the entry of the rhizobium inside the root hair as well as inside the host tissue. So through this cellulosic tube, the rhizobium proliferates and penetrates or enter into the host cortex through this root hair. And once they enter inside this host cell, The host cell is now ready for the process of the formation of the nodule. How actually these nodules are produced? The host cell start producing, or the genes which are involved in the process of nodule formation, they gets activated and they code for appropriate proteins which are necessary for the process of nodule formation. During this process, the cortical cells undergoes the process of de-differentiation. That is, the cells which have been already differentiated, they undergo a process of de-differentiation and acquires meristematic activity. That is, meristematic activity is due to the presence of various phytohormones like that of auxins and cytokinins. These phytohormones helps in rapid division of the cells, cortical cells, that is, the nodule organogenesis start. That is, nodule organogenesis means the process of the formation of the nodule is called as nodule organogenesis. This is due to the de-differentiation of the cortical cells in the cortex of the root of the leguminous plants. So once this cortical cells divide and redivide, the mass of the cells are being formed which are resembling that of a tumor. So such structure are called as nodule. Once this rhizobium enters into the cell of the host, they get they also gets surrounded by that of a membrane which are secreted by that of a host called as symbiosomes. At that particular moment, you could also see those in the images where you can find the bacterioids in the infected cells, where these bacterioids are the one which are pleomorphic and they have been surrounded by a membrane, or which has been secreted by that of a host cell. And once this membrane is on this layer is also called as symbiosomes. Once the symbiosome has been produced, the bacteria, the rhizobium, loses its morphology, become pleomorphic, and now they are capable, or they are ready for the process of nitrogen fixation. Once again, I will repeat, the rhizosphere soil of a leguminous plants, if they contain rhizobium, if there is a presence of the rhizobium, the plant also start producing certain chemical signals. The because of this chemical signals, the rhizobium are found to be specific to that of a host. And once they recognize the host, they attach onto the surface of the root hair. At the same time, this rhizobium also start producing the substance called as nod factor. These nod factor triggers the host cell to produce the proteins or any other substances which are required for the process of nodule formation. Due to the activity of the host cell, cellulosic tube or the infection thread has been formed, which leads to the formation of the process of the infection. Once the rhizobium start entering inside the root hair just curls, and through the cellulosic tube which has been produced, the rhizobium proliferates, penetrates inside the root hair and enters into the host tissue, into the cortical cells. Once they enter into the cortical cells, due to the activity of the host, due to the signals which are being reduced by that of nod factors, the host cell start producing the required substances which are required for the process of nodule formation. At that particular moment, the cells in the cortex, they undergoes the process of de-differentiation, acquires meristematic activity, and also due to the production of various phytohormones, which leads to the formation of tumor like structures called as nodule, and this process is called as nodule organogenesis. By the time they enter into the host cell, the membrane has been covered onto each of the rhizobial cells, that's called as symbiosome. And at the same time, this rhizobial cells, they are now called as bacterioids. They are also pleomorphic, and now they are capable of fixing the atmospheric nitrogen. This is how the nodule formation happen in general. Now let us study in my future videos, in next slides, how actually this nodule formation happen, which are the genes involved in the process of nodule formation, what are the product of this nod gene, what is this nod factor, how actually this nod factor triggers the host response for the production of nodule. So let us study those in detail. So now, as I was mentioning that for the process of the symbiosis between a rhizobium and the leguminous plants, both rhizobium as well as the leguminous plants, they are highly specific. They both interact with each other once once the interaction happen is due to the process of the chemical signaling molecules which are produced by that of a host as well as the bacteria. In this diagrammatic representation, we could see how actually the chemical signaling between the host plant and the rhizobium happen. So here the host plants produces or synthesizes particular substance called as flavonoids. These flavonoids are exudated or released into the rhizosphere soil. As you could see in this particular image, they are released into the particular rhizosphere soil. And the rhizobial strains are very much specific to that of a proper flavonoids. The flavonoids are the chemical signals which helps the rhizobium to see whether the flavonoid molecule is appropriate or not. If the flavonoid molecule is found to be appropriate, then the bacteria can produce the nod factor. So how this happen? Once the host cell produces these flavonoid, and once they releases the flavonoid into the rhizosphere soil, rhizobium has an ability to recognize these flavonoid molecules. How do they recognize? As we all know, the rhizobium has got a special DNA or a plasmid DNA called as symplasmid. It's also called as symbiotic plasmid. In this symbiotic plasmid, we could see a cluster of genes called as nod genes. These nod genes or nodulin genes, they are responsible for the process of nodule formation in that of a host. How it actually happen? This nod gene has got a special a gene called as nod D gene. That again I am going to tell what actually this nod gene, how actually it operate the nod genes in the production of nod factors. I will let you know in my next slide. So just remember that the rhizobium has got a symplasmid. Inside this symplasmid there are genes called as nod genes. These nod genes are the one which are responsible for the production of nod factors. Out of these genes, there is a gene called as nod D gene which code for a protein called as nod D protein. This NOD-D gene is also a regulator gene which regulates the operon of node A, B, and C. So how actually it happened? So once the node D gene produces or code for node D protein, this node D protein has an ability of determining whether the flavonoid which has been produced by the plant is appropriate or not. If it is found to be appropriate, this nod D protein and flavonoid become complexed, and they go and interact with that of a nod box, which are actually the promoter, and which triggers or which induces the transcription of nod A, B, and C gene respectively. Once the genes of node A, B, C are expressed, appropriate proteins or the enzymes are produced, where these enzymes are the one which are involved in the process of the synthesis of a nod factor, where this nod factor is also called as lipoketo-oligosaccharide. These nod factor, once they have been synthesized inside the rhizobium cell, they are released outside the cell, and these nod factors are recognized. Now nod factors are recognized by that of the root system of the host, and these nod factors influences the leguminous root to produce nodule. So once the leguminous root nodules are produced, uh, the plant has an ability of producing the cellulosic tube, where through the cellulosic tube the rhizobium cell enters into the host cell, and also because of the induction of the genes which are responsible for the nodule formation in that of a host cell, which also influences nodule organogenesis, which leads to the formation of the nodule. Once the nodules are produced inside the cells of the nodules, you could see the bacterioids which are able to fix the atmospheric nitrogen. So once again I will repeat what actually this slide depicts. The host plant produces a signaling molecules called as flavonoids. These flavonoids are recognized by the genes called as nod genes, where nod D proteins which are synthesized due to the induction of nod D gene, which is actually the regulator gene, they recognizes the flavonoids, they get complexed, and they induces the expression of the other nod genes, which are responsible for the production of the enzymes for the synthesis of nod factors. Once the nod factor has been synthesized, they are released outside the rhizobial cells. These nod factors are sensitized by the host root system, and once they have been sensitized, nodule organogenesis happen, which leads to the process of nodule formation, and also they also create an environment for the bacterial cells, that is the rhizobial cells, to enter inside the host cell, and capable of fixing the atmospheric nitrogen. So now let us study how actually this nod genes operates. As in my previous slide also, I was telling that the host specificity is determined at least two steps in mutual signal exchange between the plant and the microsymbiont. The first, the bacterial NOD genes are activated. So the bacteria, as I was mentioning, the bacteria, that is the rhizobium, they have a symplasmid. In this symplasmids, there are a cluster of genes called as NOD genes, where they have a special gene or a regulator gene called as NOD-D gene, which code for a protein called as NOD-D protein. This NOD-D protein have been activated, and they show the response towards that of a plant secreted signaling molecules such as flavonoids. The plants, if they had secreted the appropriate flavonoids, this NOD-D protein recognizes them. Once this has been recognized, NOD-D gene influences the activation or the transcription of NOD A, B, C and other genes respectively for the process of the appropriate enzymes which are required for the process of the synthesis of NOD factor called as lipocyto-oligosaccharide. And also the NOD factor which has been released from the rhizobial cells, they also stimulate or they triggers the host cell to produce the to proceed with the process of the infection by the process of the formation of infection thread as well as nodule organogenesis. So now let us study in detail how actually this NOD gene regulates. As you all know, this is just a interaction between the rhizobium and the plant to recognize the host as well as the symbiont, and also this is also a chemical signaling for the process of the infection as well as the nodule formation. How these two plants, that is the leguminous plant and the rhizobium, interacts. Once again, let us study in detail. The plants, as you could see in this image, plants produces of flavonoid molecules. The plant, if the rhizobial cells are present in the rhizosphere region, they produces a flavonoid molecules. They synthesize flavonoid molecules, and by the process of root exudation they are released into the soil. At the same time, the rhizobial cell will sensitize these flavonoid molecules. How they get sensitized? As you all know, the rhizobium, inside its plasmid, in symplasmid, they have NOD genes. Out of these NOD genes, there are certain regulator gene called as NOD-D, which is found adjacent to that of a NOD box, which we can also call it as a promoter gene. NOD-D, it's a regulator gene which produces a NOD protein. This NOD protein has an ability of recognizing a proper flavonoid. If it recognizes a proper flavonoid, this flavonoid and the NOD protein, they get complexed and they bind onto the NOD box, that is onto the promoter gene, and promotes or induces the transcription of NOD A, B, C gene respectively. And once this NOD A, B, C gene transcribe appropriate mRNA, these mRNA translates to undergo the process of translation, and they are involved in the process of the protein synthesis, and which in turn produces appropriate enzymes. Once the enzymes has been synthesized, these enzymes, they are the one which helps in the process of the synthesis of the nod factors. These nod factors are synthesized inside the rhizobial cells. These nod factors are nothing but lipocyto-oligosaccharides. You could see those in the images. They are secreted and they are released or secreted outside the cell, and these nod factors acts like a perception or the stimulus for the host cell for the where they get triggered. The genome of the host cell, the nucleus or the genome gets triggered, and they start producing the proteins which are required for the process of nodule organogenesis, as well as for the process of the production of the cellulosic tube called as infection thread, through which the rhizobial cell proliferates and enters into the cortical cells, which later gets transformed. The cortical cells divide, redivides, and they get transformed into that of a nodules, and the cells, the rhizobial cells, they establish themselves inside the cells of these nodules, and they are involved in the process of nitrogen fixation. Once again, I will repeat. Once the plant secretes an appropriate flavonoids, the rhizobial cells gets stimulated. How it gets stimulated, or how actually the recognition happen, is through the cluster of genes called as nod genes. Nod genes are found in the symbiotic plasmids, out of which there is a regulator gene called as nod D, where they recognizes an appropriate flavonoids which has been synthesized or secreted by the plant. They get complexed and they bind onto the nod box, and finally this influences the or this induces the transcription of nod A, B, C and other genes respectively for the process of transcription, translation, and the process of enzyme production, and finally the synthesis of nod factor called as lipocyto oligosaccharide. Once this lipocyto oligosaccharide has been released, these nod factor stimulates the host cell to undergo a process of or nodule organogenesis, and for the process of infection, that is the process of entry of the rhizobium into the cell of the host. This is how the nod gene gets regulated. Now, coming to this nod factors. What are these nod factor? As I was mentioning, this is also called as lipocyto oligosaccharides. This consists of oligosaccharide. Please see the structure. It consists of oligosaccharide backbone with N-acyl bond beta 1,4 N-acyl D-glucose amine, which has been bounded by a N-acyl bond. These are constructed, these are synthesized, or these are produced by an enzymes which are coded by a genes, nodule genes or nod genes called as nod A, nod B and nod C respectively. These are the genes which are involved in the process of the formation of this lipocyto oligosaccharide, which are made up of beta 1,4 N-acyl glucosamine with N-acyl bonds, where the N-acyl glucose amyl transferase, the enzyme which is being produced by nod C, they are involved in the synthesis of a tetramer or a pentamer backbone, a chyto oligosaccharide. That is, nod C genes are responsible for the production of an enzyme called as N-acetyl glucose amyl transferase. These are the one which are involved in the process of the formation of this backbone. And the nod D gene, they are the one which are involved in the process of the formation of deacetylase, where this enzyme removes the acetyl bond of the non-reducing glucosamine. You know, there are this backbone, that is chito-oligosaccharide backbone, has got a two end called as reducing end as well as the non-reducing end. The nod D gene is the one which is responsible for the production of deacetylase. This removes the acetyl group of the non-reducing. Please see how actually this, where actually this non-reducing end is being produced, is found. And nod D genes, its role is to remove the acetyl group of the non-reducing glucosamine. And nod A gene, it is responsible for the process of the formation of N-acetyl transferase. Remember, nod A gene is responsible for coding for a enzyme called as N-acetyl transferase. These N-acetyl transferase, they subsequently attaches carbon 16 to carbon 20 lipid tail to this position. So once again I will repeat, no, there are three important genes which are responsible for the synthesis of lipoketo-oligosaccharide, where this lipoketo-oligosaccharide is oligosaccharide form a backbone, where it is made up of beta 1,4 acetyl D glucosamine, which has been bounded by N-acetyl bonds. This is being coded by the genes for A, B and C respectively. The role of this node C is to code for the genes for the production of N-acetyl glucosamine aminal transferase, where they are involved in the synthesis of keto-oligosaccharide. That's the backbone. And we have a reducing end and the non-reducing end. At this non-reducing end, there are the gene called as nod B gene. They are the one which are involved in the removal of the acetyl group in the non-reducing end of the glucosamine, where with the help of, where it code for an enzyme called as deacetylase. Deacetylase, which has been coded by nod B, removes the acetyl group of the non-reducing glucosamine. That is the role of nod D. And whereas nod A, the role of this nod A is to code for an enzyme called as N-acetyl transferase, where this particular enzyme helps in attachment of carbon 16 to carbon 20 lipid tail to this position. Wherever there is a removal of the acetyl group, there carbon 16 to carbon 20 lipid tail has been attached at that particular position, which so finally this nod A, B, C are the one which are mainly involved in the process of the formation of lipoketo-oligosaccharides. And there are also various other genes like nod I gene, nod D gene, nod J genes and all. They are one which are involved in the process of the transport of this lipocoito-oligosaccharide into the outside the rhizobial cells. And also, as I was mentioning, this oligosaccharides also has got two ends, that is reducing end and the non-reducing end. Non-reducing end, as you all know, it is formed due to the modification, is modified by carbamylation as well as acetylation. This is due to the activity of nod B genes. There is one more end called as reducing end. Reducing end, they are found to be highly specific. This ends are very specific, and they are the one which decides the host, that is the reducing end confers, they helps in conferring the bacterial signals and determined by the host specific nod genes. So the role of this reducing end is found to be specific, that is for each host the this reducing ends, that is the signaling molecules are specific to that of this reducing ends. And as for example, there are certain nod O protein in Rhizobium leguminosarum where they produces calcium binding protein. Calcium binding protein which is being coded by nod O. In case of Cynorhizobium species, in case of alfalfa, they produces the nod factor that carries sulfate group on the reducing end. Likewise, we have nod H, nod Q genes, which are responsible the uh, for the specificity of the host, which confers the specificity of the host. However, finally, these nod factors are also called as lipocyto-oligosaccharides. They are being synthesized due to the genes called as nod A, B, C genes, which are involved in the process of the formation of this backbone as well as non-reducing end. Whereas in case of reducing end, that confers, that's the end which confers the host specificity. In different species of rhizobium, the genes which are present at the reducing end, they code for appropriate protein to get recognized with that of the host signaling molecules. So for example, nod O protein produced by nod O gene in case of Rhizobium leguminosarum, this is a protein which is calcium binding protein, which where it is specific only for the pea plants. In Rhizobium species, Miloti, it is they have a gene called as nod H as well as nod Q genes, where this nod H as well as nod Q gene has an ability of producing sulfotransferase or APS kinase, where these genes are the ones which recognizes the chemical signals which are produced by alfalfa plant. So finally, now we know that this nod factor is nothing but some oligosaccharide, lipocyto-oligosaccharide having non-reducing end and a reducing end. The reducing end, they are highly specific to the signaling molecules of that of a host cell that determines the host specificity. The genes which are present or involved, appropriate genes which are involved, they produces appropriate proteins, where this protein they actually confers the host specificity. So reducing end is mainly due to the role of this reducing end is to confer the specificity of the bacterial signals that determines the host specificity. This is about nod factor. So now let us study the possible signal transduction pathways triggered by the nod factor in the leguminous root cells. In my previous slide, we studied how actually these nod factors are produced, what are these nod factor, which are the genes which are involved in the production of this nod factor. As I was mentioning, nod factors are the one which acts like a signaling molecules for the process of the triggering of the host, for the process of the formation of nodule, as well as for the process of the entry of this rhizobium inside the host cell. So how actually this signaling transduction happen? Let us study in detail. So if you concentrate on this particular image, you could see the nod factor, which have been recognized by the receptors which are present on the host cells. These receptor like kinase which are involved in the process of the recognition of the proper nod factor. So once the nod factor has been recognized by that of a host, there is a process of calcium. Once this happen, early nodulation process, certain events happen. Which are those? That is, the there is an activation of calcium influx into the cytoplasm, and calcium spiking in the perinuclear region happen. This has been mediated by certain genes called as castor or pollex and LUPI 33 133 gene, LUP 133 genes. The products of these genes are or the formation of ion channels. Castor or and pollex genes, they are actually the twin genes. They are actually the twin homologous genes which are present in Lotus japonicus. The main role of these genes are to encode for ion channel protein. Through the formation of this ion channel, there is an influx of calcium inside the host cytoplasm, and also calcium spiking in the periplasmic region, perinuclear region happen. Thus calcium is the one which triggers the certain genes called as calcium calmodulin dependent protein kinase. And also there is one more gene called as DMI3 genes. DMI3 gene and CCAMK, these genes are activated, and which in turn they helps or they induces the transcription of genes called as NSP1 and NSP2 genes. These are NSP1 and NSP2 genes are the one which are involved in the process of the formation of nodules. So once they express, such genes are being expressed and such gene transcription and translation of the protein happen, these proteins are involved in the process of the formation of cellulosic tube, as well as for the process of de-differentiation of the cortical cells, which leads to the formation of a mass of cells, maybe also due to the phytohormones which play a major role in the formation of the nodule. So this is the signal transduction pathway for the process of the triggering of the early nodulin genes which are involved in the process of nodule formation, as described by Marico et al. In 2006. And this is also one of the model which has been illustrated by Christopher et al. In 2012. This is the schematic illustration of the genetic components involved in legume rhizobium symbiotic signaling. So how actually the signaling happen between the leguminous plant and the rhizobium has been illustrated by Christopher et al. In 2012. According to this model, the nod factor which has been synthesized by the rhizobial cells or perceived, that is they are perceived by the or they the signaling sorry the receptor complex which are present on the surface of the host cell, they perceive this nod factor, and due to the perception, what happens, the perception of this nod factors triggers the early response, like influx, ion influx, that is calcium influx, depolarization of the plasma membrane, production of reactive oxygen species, and the cytoplasmic alkalization happen. Because of the perception of the nod factors, the early response are like this: ion influx, depolarization of plasma membrane, production of reactive oxygen species, cytoplasmic alkalization happen. Later, the signals and the transducted downstream to activate LRR receptor kinase. LRR is nothing but leucine rich repeats. Okay, these receptor kinase and DMI2 and DMI1. These DMI1, DMI2, they are involved in the process of the formation of ion channels, and which is required for the process of calcium spiking. That is influx of calcium and calcium spiking around the perinuclear surface. Later, these calcium signals are perceived and decoded by calcium or calmodulin dependent protein kinase and DMI3, which acts as a central player in the nodule factor signaling and coordinate the expression of symbiotic genes, including early nodulin genes, and later nodulin factor signaling culminates with the coordination and synchronous progression of infection and the cortical cell division, resulting in the formation of mature infected and nitrogen fixing nodules. So in this particular image, you could see the node factor which has been synthesized by the rhizobium, and they are pursued, that is they are pursued, they have been stimulated, or they just bind onto the receptor proteins which are present on the surface of the host cells. Once they have been pursued, there are certain early responses which happens, that is the influx of iron, depolarization of plasma membrane, and even the production of reactive oxygen species, and also the cytoplasmic alkalization happen. These are the early response because of this perception. Later what happened? And also this influx of this calcium and all happens due to the certain genes called as DMI2 as well as DMI1. These are involved in the process of the formation of the proteins which are involved in the formation of this ion channels, where they create an ion channels into the nucleus, because of the calcium spiking, because of this calcium signals which has been produced inside the host cytoplasm as well as inside the nucleus. There are certain genes called as DMI3, these, and also calcium or calmodulin dependent protein kinase enzymes which are present, and also the genes which are involved in this DMI13, even also this also are involved in the process of the formation of ion channels into the nucleus. This triggers, or this enzymes activates the genes which are present inside the host genome, which are responsible for the process of the formation of respective amino acids, or the protein and the enzyme for the process of the expression of for the process of the formation of this infection thread, as well as for the process of the formation of the nodule and the nodule organogenesis. So this is about the legume and the rhizobium interaction and the process of nodule formation. And these are some of the references, and thank you.